Welcome to the Anatomy Dissection Laboratory for students. In this video, we will learn how to dissect the nasal cavity. Dr. Carlos Suarez-Quian and Joel Valensky are the authors of this presentation. Dr. Suarez-Quian is the narrator. The nasal cavity is the primary entryway for air into our body to eventually reach the lungs. Mucosa in the nasal cavity warms and moistens the entering air. Unfortunately, it also is the primary entry for microorganisms to enter the body, including COVID-19. Let's start the dissection of the nasal cavity. In the previous lab, we bisected the head, and as recommended, the bisection was done so that the cut was a little off-center to preserve the nasal septum. Let's identify a few structures to become oriented with the image. Here we see the frontal sinus, the hard palate, the tongue, the uvula or soft palate, and the body of the sphenoid bone. Now that we are somewhat familiar with the image, we can say that we're looking at a wall of the nasal cavity, specifically the nasal septum. The nasal septum represents a medial wall of the nasal cavity. Hence, in this image, one nasal cavity is deep to this medial wall. The nasal septum consists of bone, the vomer, which means plow in Latin, and indeed resembles a plow, the perpendicular plate of the ethmoid, and the septal cartilage. The vomer, perpendicular plate of the ethmoid, and the septal cartilage must come together during development to help form the nasal septum. It is no wonder that the septum is generally deviated in people because it is difficult for the bones and cartilages to come together in one plane. And just to convince you that there really is a cavity behind the septum, we can look at the coena, the back passageway that connects the nasal cavity to the nasopharynx. Air, indicated by the wavy yellow lines, passes through the coena on its way to the lungs. Do not confuse the coena with the nasal concha, which will be seen in the next image. At this point, the nasal septum needs to be cut away to demonstrate the lateral wall of the nasal cavity. However, before doing so, look for the nasopalatine nerve deep to the mucosa at the level of the vomer. Realize also that we do not see the olfactory nerves in the nasal cavity. They are microscopic. But be aware that the olfactory epithelium, that is, where we respond to smell, is just inferior to the ethmoid bone. Once the nasal septum is removed, the lateral wall of the contralateral nasal cavity becomes apparent. Thus, we're looking at the wall of the left nasal cavity. The three protruding structures seen here are the three nasal conchae. These bony structures are covered by mucous membrane and cilia and act to moisten and warm inhaled air. Inferior and lateral to each of the conchae are recesses, called meatuses, into which paranasal sinuses drain. The largest sinus, the maxillary sinus, drains into the middle meatus. The nasolacrimal duct, meanwhile, which conducts tears from the eyes, drains to the inferior meatus. The auditory tube runs from the nasopharynx to the middle ear cavity and allows for the equalization of air pressure on both sides of the tympanic membrane, or eardrum. The tube is only open during swallowing or yawning, and that is why we swallow when ascending or descending in an airplane. The opening of the tube is guarded by an elevation known as the torus tubarius. Recall that children have relatively larger and straighter auditory tubes than adults and have a large mass of lymph tissue, pharyngeal tonsils, overlying the auditory tubes. When these pharyngeal tonsils become inflamed, they become known as adenoids. The arrangement of the auditory tubes are factors that explain why children are so susceptible to middle ear infections. Again, the olfactory epithelium, which contains the sensory neurons responsible for smell, is located high in the nasal cavity. This location explains the loss of smell being an early symptom of COVID-19 infections. The virus infiltrates this region and impedes the transmission of impulses from the epithelium through the cribriform plate to the olfactory bulb. In a different cadaver dissection, the different appearance of the concha can be appreciated. The concha will vary in size from individual to individual. Removal of all three conchae reveals the ethmoid air cells, which are located between the posterior aspects of the eye orbits. This arrangement accounts for the spread of eye infections to these air cells. The hard palate and location of the orbit are indicated in the image to orient you. The location of the inferior concha is now indicated. The ethmoidal air cells are now clearly labeled. The use of the term cell is often confusing for some students as they assume we are describing cells one sees with a microscope. In the present context, the term cell is being used to denote a chamber, that is, a region surrounded by walls. An opening below the inferior concha is now labeled the opening of the lacrimal duct. This is where tears from our eyes travel to when we cry. Moving to the area where the middle concha was located, we see an elevation called the ethmoid bulla, and just below it, a space called the semilunar hiatus. The opening for drainage of the maxillary sinus is within the semilunar hiatus and is located high relative to the sinus, which can impede sinus drainage. To enable better drainage, surgeons may make an additional drainage port lower on the sinus wall. Further dissection into the lateral wall of the nasal cavity leads to the maxillary sinus. For orientation purposes, the hard and soft palate are labeled, as is the orbit. In this image also, we can see the perpendicular plate of the palatine bone covered with mucosa. If we remove the mucosa and the bony vertical plate of the palatine bone directly posterior to the maxillary sinus, we see a canal that conducts the greater and lesser palatine nerves arising from the pterygopalatine ganglion. The nerves will supply the hard and soft palates, respectively. It has been speculated that ice cream headaches are due to the cold affecting the cells in this ganglion. The pterygopalatine ganglion receives sensory innervation from the maxillary division of the trigeminal nerve. At this location, the nerve is running on the roof of the maxillary sinus and becomes known as the infraorbital nerve. In a different dissection, the relationship between the infraorbital nerve, pterygopalatine ganglion, and the greater and lesser palatine nerves is demonstrated. This now concludes the dissection of the nasal cavity.